My project would be able to save the grouper fish, which is a very important fish within our region and in Singapore. First infected the cells with virus, then we injected the chemicals into the culture flask. Then we investigated how well the chemical inhibited virus replication. So from there we compared the effectiveness of the two chemicals and we made a comparison.
I'm trying to find a relationship between the mathematical and the physical aspects of chaos. It opposes our normal thinking of randomness, and chaos actually proposes that there's no randomness, although we still cannot predict to our future to infinitely long.
Two physics projects. The one that you see behind me deals with plasma physics and the deposition of titanium nitride on stainless steel. And my second project at MIT, it deals with the magnetism of this compound called the vanadium geroside. The main use of titanium nitride is in tooling industries and in electronics so on, to extend the life of your tools because it's very hard, so when you coat it on the stainless steel it can sort of protect it. I took up this project because it helps to enhance the engineering performances of metals, and I believe that this will be very beneficial to many industries.
Trying to come up with a new technique to diagnose the type of dengue virus infection. I thought that doing something that related to humans was most intimate and most satisfying. I guess I could go as far as I would like to zero down my research to helping people.
The objective of my research is to discover a vaccine for the Epstein-Barr virus. By using a computer scoring system, you can find out the interactions of the Epstein-Barr virus with the immune system of humans. In future, using more results and more experiments, derive particular vaccine for the virus and counter its effects.
The objective of my project is to investigate the effects of chlorofibrate on red hepatocytes, which is red liver cells. And to do that, we introduce chlorofibrate into red liver cells, and we measure several factors like oxidative stress, membrane potential, to investigate the mechanisms by which the drug works on the red hepatocytes. Chlorofibrate is a hypolipidemic drug that is widely used and is found to have the side effects on red hepatocytes. So we're basically just investigating these minor side effects on the red liver cells, and that might have future use in medical fields.
I want to design a faster and more efficient method to look for natural antifouling compounds that will not have environmental legacies and will be environmentally friendly.
Biofouling is just basically the attachment and growth of living things on a submerged surface. This includes very small creatures like bacteria and other larger creatures that can be seen like barnacles. When into biofouling, I realized that it was a serious problem without a proper solution. Yes, because at present people use toxins which obviously have environmental legacies. So I decided to go into this research to design a new method to look for some biodegradable compounds.
I wanted to learn more about audio compression methods, and if possible to come up with more efficient methods of audio compression to achieve a higher rate of compression ratio. Ultimately, it's the brain and the human brain that perceives the sound. So if there's some information that do not need to be coded, then what for code them and then waste storage space, you see?
I've always been fascinated by 3D stuff, and I thought it would be really cool to convert a real object into a 3D object in the computer. For the project, we need two images to find the coordinates of one point. It's actually a new branch of virtual reality, you see, because you can actually generate a real object into a 3D object in the computer.
Duchenne muscular dystrophy is caused by a defect in the dystrophic protein. So it's either the protein is not there or it's there but not working. It's fatal because it affects the muscles, you see, and your heart's a muscle.
So for methods such as drug design, it's quite a novel way of doing things, in the sense that you visualize the thing first before you design the drug. Instead of using by theory, you think of something, then you do something else, then you visualize, then you see whether it's compatible, reject it or approve it, then go ahead.
You want this one, huh? Okay, then we move this one. 1, 2, 3, 4, 5.
Even though muscles is a very vital part of our body, it's not a well-studied part. There are many restrictions when it comes to studying muscle tissues. And personally, I'm a sports reporter at Taobao. So I am very interested in knowing how sportsmen actually function, like how they run or how they actually perform to their best. Knowing the mechanism of this process, we'll actually be able to devise drugs such that it will be able to slow down this process or inhibit this process so that we will be able to allow patients to recover from muscle spasm more quickly than we actually are able to now.
I wanted to find out if glucosamine is actually a really effective drug for treating arthritis, as it's commonly believed that it is the cure for arthritis, but people don't really know what actually happens when you take glucosamine. Glucosamine actually stimulated the formation of proteins and proteoglycans in these cells, but this stimulation was actually only for a short period of time, contrary to popular belief, and it actually inhibited the synthesis of these substances after 6 days, which is a longer period of time. And the interesting thing about it was that we observe a dose-dependent response, which implies that glucosamine has some kind of effect on the cell's metabolic pathways.
I study an enzyme which has several functions in the human body's immune system, and basically what I did was to study the relationship between this enzyme's location on the cell membrane and how it functions over there. The excitement of finding things that had never been discovered before in the world, that after all my reading into this protein and its functions and what it does and all the experiments previously done on it. And yeah, I found in the course of my experiments I did find some things that have never been reported before in the course of research on this protein. So that's why I actually got quite excited about my results, and it opened up a lot of new questions which I'm going to answer. The hope for my findings is that firstly they'll be ratified as being correct by work done by other people, and in addition to that I would also love to see my work actually being used as the base from which more experiments can be carried out.
More than 50% of the fatal snake bites in this region are caused by the Malayan krait, and it's among the 10 most poisonous snakes in the world. So if I could manage to come up with something on it, we could actually find a cure for the snake bites. What we're actually doing is we're adding on to the literature, which is the pool of all this data available to them, so that one day maybe they could be able to use it and find the universal cure.
Just like in the case of fencing, timing is everything in the case of the Malayan krait. Not only is correct identification of the snake crucial, but after the snake has been identified, more than 50% of the snakebite victims still die even with the correct antivenin. We hope to be able to find a more potent antivenin for the Malayan krait.
Well, basically what we've done is we've taken this venom out of the snake so that now we can produce it in the laboratory without having to go back to extract it from the snake. And from there, you can branch off in a whole lot of other things.